Trinity Cultural Academy, Coimbatore, and New India Publishing Agency, NEPAS, New Delhi, welcomes you all for this presentation on Principles of Seed Production, Part 2, Tidbit 16, which comes under the major subject Plant Science and Seed Science and Technology. And this material is highly useful for ICAR All India Entrance Examination for Admission JRF and SRF and ICAR NET ARS STO and IBPS AFO examination. And this material is prepared by Dr. K. Vanangamudi. He is a formerly Dean Agriculture, TNAU Coimbatore, and Dean Adiparashakti Agriculture College, Kalavai, and Professor and Head Seed Science and Technology, TNAU Coimbatore. And so far in the previous video, we have seen the agronomic principles of varietal seed production. So in this video, we are going to see about the hybrid seed production. In that, what are all the major principles we are following? That and all we are going to see. And behind this, we are going to see genetic principles of seed production also. So let us see one by one in detail. In case of hybrid seed production, all the operations are similar to the varieties except certain following steps as given below. The first one is planting ratio. Planting ratio, it may vary from crop to crop depending upon their seed production capability, and it is nothing but the ratio to be maintained between the female and male line, not male and female line. The female ratio should be higher because it gives us grain or seed anything. So female and male lines in order to have a good amount of pollen grains for proper pollination and fertilization. So the planting ratio recommended for particular species should be followed. And for example, in case of rice, it is between 8:2 to 10:2, 8 rows of female and 2 rows of male, or it may be 10 rows of female and 2 rows of male. In case of maize, it may vary between 4:2 to 6:2. And in case of sunflower hybrid seed production, it is 4:1. Mostly the planting ratio is followed for hybrid seed production purposes. And in case of cotton, a special system called block system is followed. Here the female and male are raised in the ratio of 8:1 in separate blocks, but not more than 5-meter distance between these two blocks. So this will aid in easy identification of male plants and aids in pollen collection, since hybrid seed production is by emasculation and dusting. We are going to see about what is emasculation, what is dusting, at what stage it should be done, that and all we are going to see in hybrid seed production of cotton. Now we see just about the what is emasculation. It is nothing but a removal of male part of the flower, and dusting means the pollination behavior, like that. Here we can see the difference between male and female rows. Here these are the male rows and these 4 rows are the female rows. So female rows are higher compared to the male rows. In case of this is in case of maize, this is in case of rice. Here 8 rows of female rows are planted and 2 rows of male lines are planted here. And the second important aspect is that border rows. This is also very important. The seed field should be planted with border rows in all these 4 sites with male parent seed, not female parent. The 4 sides of the field should be planted with border rows of male parent seeds to have more pollen source and also to avoid contamination. The number of rows, border rows may be from 4 to 12 depending upon the crop. So here we can see that this is the first border row, and this is the second, and this is third, and this is fourth. It may vary from crop to crop. Here in this case, this is in case of maize, 6 border rows are followed. It may vary from crop to crop. And the next important practice of hybrid seed production is synchronized flowering. This is to having the uniformity of flowering between the parental lines, and to achieve this uniformity of flowering, any one of the following techniques can be done. So one is staggered sowing. This is nothing but adjusting sowing time to adjusting the sowing time of parental lines to have a synchronized flowering habits, and withholding irrigation and application of DAP or urea to enhance the flowering habits. Let us see one by one in detail. In case of staggered sowing, it is nothing but adjusting sowing time of parental lines in order to have a synchronized flowering between the parental lines for proper seed set. For example, early flowering line should be sown later, and late flowering line should be shown earlier. For example, in case of sunflower, KBSH1 hybrid seed production, male parent is late flowering, so it is late flowered by 6 days than the female. So sowing of male line 6 days earlier than the female line ensures synchronized flowering. And the next one is in case of rice, exertion of female panicle from boot leaf is a major problem. To have a complete exertion, application of GA3 gibberellin at 70 to 75 gram per hectare twice during flowering time enhances the exertion of female panicle, since GA is a cell's elongation factor and it aids in exertion of panicle. And the next special techniques which are followed for hybrid seed production in for a specific purpose in different crops are: first one is detasseling. It is nothing but a removal of tassel, removal of male part of the flower from the female row is nothing but a detasseling. It is followed in case of maize. Here we have given the picture. This one is the tassel, and it should be removed in case of maize hybrid seed production. And the next important is emasculation and dusting. This is followed in case of cotton. Emasculation is nothing but removal of male part of the flower, and dusting is nothing but the dusting of pollen to the female organs. So it should be done at proper time of flowering to have a good yield and to avoid contamination. And this technique is not followed in case of crops where the hybrid is produced by utilizing male sterility system. It is very easy because manual practices are not used there. So example here is sunflower. And the next important factor is supplementary pollination. It is that improving the pollination behavior to have a good seed set. And in case of rice, rope pulling or rod driving is practiced. Let us see clearly in rice hybrid seed production. Here you may know about only what is supplementary pollination. What is the technique followed in rice? Is nothing but a rope pulling or rod driving. In case of sunflower, rubbing or installation of beehives is a supplementary pollination technique followed in case of sunflower. So rope pulling in case of rice is followed. Here the two persons across the field are pulling the rope against the crop. And the next important factor is genetic contaminant. The contaminant may be physical or genetic. So genetic contaminant in case of this one is, first one is off type. This we have seen in the varietal seed production also. This is nothing but the plant which is other than the seed crop is nothing but a off type. This is a deviant of seed plant. It is common for both hybrids and varieties. And pollen shedders is nothing but a presence of B line in A line is in hybrid seed production. Example here is rice, particularly in hybrid seed production field. And this one is very important. What is um presence of B line in A line is called as DAFs. It may ask an exam. So it is very important point. And the next one is pollen partials. It is observed in case of bajra. And shedding tassel. Tassel we have know that this is deals in case of maize. This is nothing but presence of tassel in female row. And selfed bolls we have know that it is in case of cotton. Selfed bolls may occur without doing emasculation and dusting process, and selfed fruits in case of other vegetables. And the method of harvesting is also very important factor, because in case of hybrid seed production we are using more than two lines, A line, B line, R line. So it should be harvested separately and packed separately and threshed, etc. All the post harvest process should be carried out separately in different plants. And in case of hybrids, harvest R line first. This is a restorer line, and remove produce from the field and carry out all the operations, or and it should be done in separate field. And another one is do final rogging in female row and do harvest. And so far we have seen the varietal seed production and hybrid seed production, and what are all the agronomic principles followed in case of seed production aspects. And now we are going to see about the genetic principles of seed production. This is also we have seen separately in hybrid and varietal seed production. Even though we have seen that, let us see uh, in overall point of view. The first one is there are two scientists explained about this genetic principles of hybrid seed production. The first one is Home in the year 1953, and Hartmann and Kester in 1968, maybe wholly or in part to maintain high levels of genetic purity during hybrid seed production, is suggested by these authors. And the various steps suggested by Home in the year 1953 to maintain the varietal purity are use of approved seed sources in a seed multiplication process, and inspection and approval of field prior to planting, and field inspection and approval of growing crops at critical stages for verification of seed genetic purity, reduction of mixtures, weeds, and freedom from noxious weeds and seed-borne diseases, sampling and sealing of cleaned lots, and growing of samples of potentially approved stocks for comparison of with authenticated stocks. And the various steps suggested by Hartmann and Kester in the year 1968 for maintaining the genetic purity are as follows. Here, the major one is providing adequate isolation to prevent the contamination by natural crossing or mechanical mixtures, and rogging of seed field prior to the stage to which they contaminate the seed crop, and periodical testing of varieties for genetic purity, and avoiding genetic shift by growing crops in areas of their adaptation only, and certification of seed crops to maintain the genetic purity and quality of seed, and adapting the seed generation system for based on the pollination behavior, and grow test. This is conducted to determine the genetic purity of the seed. And the various important safeguards for maintaining the genetic purity during seed production are these points are also we have already seen in case of hybrid seed production. Let us see one by one. The control of seed source, the use of appropriate class of seed and approved source is necessary for raising the seed crop. Starting from breeder's seed, the seed can only be multiplied up to 3 more generations as foundation seed, registered seed, and certified seed. And for four classes of seeds, we have know that breeder, foundation, registered, and certified seed. So in this system, the seed production is restricted to four generations only, depending upon their pollination behavior. And the next one is preceding crop requirement. This has been fixed to avoid contamination through the volunteer plants and soil-borne diseases. And the next one is isolation distance. This we have seen in the previous video in varietal seed production. Clearly there are three types of isolation: barrier, time, and distance isolation. This is to avoid the natural crossing with other varieties which are grown along the side, or off types which are present in seed field. And it may be due to mechanical mixtures at the time of sowing, harvesting, threshing, processing, etc. So next one is roguing. Roguing, we have already seen that the removal of rogue. Rogue is nothing but the existence of off-type plants. The removal of off-type plants is called roguing. So the plants which are differing in characteristics from those of the seed variety is another potent source of genetic contamination. There are three main sources of off-type, maybe due to some recessive genes in heterozygous condition at the time of release of varieties, or it may be due to volunteer plants which may arise accidentally from planted seed or from earlier crops, or it may be due to mechanical mixtures during the process of post-harvest operations. And the next important aspect is that seed certification. So, the genetic purity in commercial seed production is often maintained through a system of seed certification. We are going to see in further videos about the seed certification detail, but here we are going to see little bit few points. That is, the principal objective of seed certification is to maintain and make available crop seeds, tubers, and bulbs, etc. And here the qualified, well experienced person of seed certification agency carry out the field inspections at appropriate stages of growth for different crops as different times at different times. It may vary depending upon the pollination behavior. That is, the number of inspections may be vary depending upon the pollination behavior. For cross-pollinated crops, it is high, and in case of self-pollinated crops, the number of field inspection will be low. And also make seed inspections to verify that the seed crop or seed lot is of requisite seed genetic purity and quality, and after harvesting to verify quality at the time of processing, and draw samples for seed testing and sometimes for grow out test also. Specific test to determine the genetic purity of the seed is nothing but a grow out test. And in addition to inspection, seed certification agency also lay down some standards for in a field and also for seeds, to which the seed crop and seed lot respectively. To the seed crop, field standards is prescribed, and for seed lot, seed standards is prescribed. What I mean by field standard and seed standard means, field standard, it is nothing but it is maintained in case of field in field aspects, that is the land requirement, isolation requirement for particular crops, and maximum permissible limit for off type and shedding tassel in case of hybrid seed production, etc. And in case of seed standards, it is nothing but a germination percentage, moisture percentage, and physical purity, genetic purity, etc. And seed certification implies that both the seed crop and seed lot have been duly inspected. And we have already seen that the grow test is periodically tested to determine the genetic purity, and the varieties being sown for seed production should be periodically tested, and this is to make sure that they are being maintained in their true form. And thank you friends, thank you for watching this video. And in future videos we are going to see the individual crop seed production techniques one by one. Thank you for watching.